Introducing the PhyNexus AutoPlasmid MEA for hands-off biosample prep. It is a multi-tool benchtop robotic system able to process 1 to 12 samples at a time in plates of up to 96 samples. The sample preps that are able to be performed include Mini and Midi plasmid purification, mouse tail genotyping prep, direct from culture plasmid prep, affinity protein chromatography, prep and scale up affinity process development, protein affinity, protein engineering, desalting buffer exchange, ion exchange chromatography, on-column enzyme assays, and more. The instrument is based on a 12-channel head, to which pipette tip columns are attached. 96 samples are processed 12 at a time, moving across 8 rows on a plate. Shown here are 3 types of columns that may be operated on the instrument. The instrument is based on dual flow chromatography, a proprietary technology of PhyNexus. With dual flow chromatography, fluid flows back and forth through the pipette tip column. Interactions of plasmids, proteins, or other biomolecules with the column are pushed to completion even with a limited sample. Through this fluid motion, columns are completely loaded, allowing high recovery of the sample. This view is looking down at the deck. Setup time is 5 to 15 minutes depending on the application. Columns are loaded on the left of the deck. Samples are processed moving to the right of the deck. Samples are recovered at the end of the deck. In this deck layout, plasmids are purified directly from the cell pellet. Either mini-scale or midi-scale plasmid preps may be performed. In this deck layout, 288 purification conditions were studied parallel method development for purification of an antibody. The resin loading, washing, and elution conditions were studied. Yield was measured by ELISA and protein activity was measured by SPR. The results showed that it is possible to achieve high yield while retaining the clone protein activity. But the results also show why it is important to screen purification conditions. It is possible to combine processes to be operated in series automatically. In this deck layout, recombinant proteins are first purified by IMAC affinity chromatography. After recovery of the proteins, the samples are directed to a gel filtration column for buffer exchange to remove imidazole. Because of fine control, the samples are not diluted. Proteins remain at high concentration even after 2 operations. The AutoPlasmid MEA eliminates the tedium of manual procedures by performing all the necessary steps automated and in parallel. Step-up time is rapid, and up to 96 samples may be processed depending on the application. See your PhyNexus representative for more information. Thank you for your attention.